At the time of registration, our audience members have the opportunity to submit questions for us to consider. And this next one, Donna, I'd love for you to connect with this. So is there an approach to reducing batch-to-batch variation of cell therapy products, particularly critical quality attributes or CQA, such as potency?
Yes, most definitely. I think one of the things, a lot of the cell and gene therapy development comes out of academic R&D research aspects. And while that's critically important, there's that migration to translational and obviously early to late stage GMP manufacturing. What becomes important is something that I think gets ignored is even if it's in its infancy, is understand that target product profile. And it's not something that gets discussed very often, but it's like, what is the product? What do you expect it to be? What is your proposed mechanism of action? And how do you intend to monitor that? What are your expectations? So early on, that's what you have to start with. As you go through manufacturing, as you migrate through your clinical trials, whether you're, and even as you work with healthy donors to garner all your CMC regulatory work that you need to do, and understand and perfect your manufacturing process, you can gain a lot of information about what to expect and how to best monitor that going forward. So I think, we always start with these are the critical quality attributes. This is what we expect in our product. Once you get more and more into that, you can tighten those expectations as your data set increases. And for me, that whole quality by design is a huge impact on product manufacturing for cell and gene therapy. It also helps control that variability. It includes not only the manufacturing process and the variation there, it includes your reagents and consumables, more so your reagents, to assure that you don't have variability in those reagents that play a role in your outcome. But as you progress from early stage translational development and scale from R&D to GMP manufacturing, the use of the right reagents that are acceptable for manufacturing. So you start early with those, and you also make sure that you have the means to transition from one reagent to the other. If you start with a non-GMP and you transition to GMP, what are those expectations, and make sure that that GMP reagents meets those same expectations. I like to use the example in the early days of cytokines. There's tons of R&D cytokines that tend to, in general, have a lot more potency than the GMP cytokines that you use in manufacturing, for the simple reason they don't have as rigorous a purification process at the end. So understanding all those contributors is important, making sure that you control all the aspects that you can and understand their role in the critical quality attributes in your process development. So defining those critical attributes, defining the contributions to that. And so it's including of your mechanism of action, your process knowledge, and understanding the impact on the outcome of the final product is hugely important. It includes the understanding your seed materials and all your critical components that go into your manufacturing.